Oh, good morning.
Hello.
Yeah,
Can you hear me?
Yes, I can hear you and I can see you. And my video is not active yet. I don't know what our teams are doing, what are they doing? They're censoring us. All right, hopefully they will it will activate the video. Let's see. Yes,
is the audio working?
Yeah, I'm up there. Good morning, sir. Thank you
Hi,
very much.
AJ.
We are so
I can't
grateful.
hear you. Can you hear me?
We are really honored to have you. Wonderful, beautiful. All right, so that's we are audio active, video active, and all right, your screen is shared. That's fantastic. Yeah, wonderful. That's beautiful.
I can't hear anything.
Well, you really become an expert on this. You have done thousands of times.
There you are.
It took no time for you.
Now I can hear you. Can you hear me?
Yes, sir. That's beautiful.
Okay.
Yes.
We're all set. I think we're all set then. Okay.
That's fantastic. All
okay.
right.
Good. All right. Well, I'll go get my glass of water.
Okay.
And we'll be ready to go at 9 o'clock. Okay.
Great. Beautiful. Absolutely.
Thank you very much for inviting me for this.
It's an honor for us, sir. It is indeed an honor for us. You are one of the most eminent scientists, physician of our epoch, truly are. I mean, you have made fundamental, and this story that you're going to present indeed is beautiful to see how from the very early days of saying atherosclerosis and inflammatory disease to come really to full head. And now we have interventions that showing that targeting inflammation indeed attenuates the phenotypic expression of atherosclerosis with clinical outcomes that are meaningful. So we are really seldom happens in life, as you very well know, as the leader, that one can take it all the way from very basic fundamental immunology of it to clinical application of it. That's beautiful.
Yeah, well.
So, by the way, couple of things. Klaus Levy will join us a little late. He is running grand rounds from his time 8 to 9, so he will join a few minutes after 9. And Jake Lucis sent his regards. He unfortunately has COVID.
Oh dear.
And his voice is still gone. Yeah, so he couldn't join us. He just texted me last night late saying he waited to the last minute, but unfortunately he can't join because he still is not feeling well. So he did send his regards to you.
Okay, and you have
Valentine will be joining us as Christy Valentine and the others, as indicated before, they will be joining us as well.
So Matthias
Hansen had a commitment. He did say to give his regards to you, and he won't make it either. So wonderful to have all these people really appreciating you and expressing their gratitude to what you have done over the years.
Okay, great. So is Matthias Nahlendorf going to be online?
Who will be?
Matthias Nahlendorf?
Yes. Matthias will be online. He is joining us as a panelist.
Okay, good.
And then who else will be here as a panelist? Christy and Yibing Wang, who is one of our associate editors. He was at UCLA. He recently moved to Singapore. He will be joining us as a panelist as well. And let me look at my list and see who else I had. I think the journal is, as in like any other new journal, at the beginning is a struggle. There is, 30,000 biomedical journals, so it's not easy to make headway. It's amazing, but it is. I think it's getting some traction now and gradually getting some attention. I think we are publishing reasonably good papers, and so we try to make focus on quality as much as possible, so we don't get recognized as a junk journal. That's a key issue when you start the distinction to be made, and I think we are gradually making some headways, and I think this year we should be able to get listed in some of the citation indices, and hopefully gathering some citations. As you know, we authors are the slave to impact factor. I mean, people put the papers, journal ranking in front of them, and then go where to publish based on impact factor. Hopefully, that will gradually will be shying away from that approach and focusing on the content and scientific contributions rather than IF of the journal. So. We have, I think we still have struggling attracting original research articles. And I think once we have a few good papers published and review papers are published, hopefully we should be able to make that transition and hopefully, receive some good quality original research articles.
Good. All right, well, I'm going to go get my glass of water and get mentally prepared. Okay, so I'll be back with you in just.
All right. Any others? See, panelists are. Let's see. We have Mhm. 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 All right, it's precisely 8 o'clock. Dr. Libby, do I have your permission, sir, to start?
Of course, you don't need my permission. You're the boss here.
Thank you, sir. I'm honored. So good morning, everyone. And we are delighted to welcome everyone to our JCA seminar series. Today, it's my distinct pleasure and great honor to introduce and host Dr. Peter Libby, who is a world-renowned physician scientist. He is the speaker for our webinar today, which is named after Dr. Eugene Braunwald. And this is Eugene Braunwald, MD lectureship. Dr. Braunwald is considered the father of modern cardiology and has mentored numerous leaders in the cardiovascular field. And Dr. Libby also was mentored by Dr. Braunwald. And so the lecture is named after him and is the most fitting that Dr. Libby, the most prominent scientist of our time, to give this lectureship. This lectureship is part of the JCA webinar series. JCA is a relatively new journal, and which, let me share my screen with you briefly. Which is entering about 2 years of publishing. And it is an open access journal. We don't charge anyone for publication in JCA, at least for another year or so. And after that will be the conventional fee. It is likely one of the fastest way to get your paper published. We are interested in broad cardiovascular aging category. We really not narrow focus on cardiovascular aging, but everything related to the process of aging from birth to death fits into the journal. We have distinguished board member, including our speaker today, Dr. Peter Libby, who are always been amazingly gracious and wonderful supporter of all academic activities, globally, and leadership in this also. We're truly grateful to him. The journal has in the second year of publishing and gradually getting traction and getting some attention. And we have published 72 articles, 40 of them which are listed in PubMed PMC, and we anticipate to get listed in citation indices this year, including PMC and PubMed, and hopefully we'll be gathering impact factor soon. We have initiated a number of efforts at the JCA, including webinar, and Dr. Libby will be giving today's webinar, which is Eugene Braunwald, MD lectureship. Dr. Peter Libby is indeed one of the most eminent physician scientist globally. He received his MD from the University of California, completed his training at a Harvard system, and as a resident, including his residency fellowship as well as research fellowship, where he became a faculty and soon rose the rank of academia, and took the helm of the cardiovascular medicine at Brigham and Women's Hospital, where he was chief of cardiology for over a decade and established the program, one of the most distinguished and reputable program in the country in training, as well the future leaders in cardiovascular medicine. Dr. Libby currently holds the position of Mallinckrodt Professor of Medicine at Harvard, and he has numerous leadership role at the Harvard system, as well as outside that I will mention briefly. From the very outset, Dr. Libby focused on delineating the immunology of atherosclerosis and inflammation. This was a journey really that took him from basic fundamental discoveries ultimately to clinical trial targeting interleukin-1B with a monoclonal antibody in the famous CANTOS study that really ultimately culminated in establishing inflammation as a causal factor in atherosclerosis. His seminal work has been published in all major journals from New England, The Lancet, and Nature Medicine, and PNAS, and others. He has published over 1,200 articles, which have received over 250,000 citations, and has garnered him an H index of above 120, which is really one of the most, Dr. Libby is one of the most cited investigator in cardiovascular field. Indeed, this year also he was selected as the highly cited investigator of the year. In addition to his leadership position at Harvard System, Dr. Libby has served in numerous leadership positions from NIH, AHA, and ACC. Currently is the president of International Atherosclerosis Society. He was the chief editor of the most prominent textbook in cardiovascular medicine, Dr. Braunwald's textbook of heart disease. It remains one of its main editors currently. His research has been funded by NIH as well as the Leduc Foundation and Reynolds cardiovascular program, which has supported program in a very few selected centers in the country. For his recognition, Dr. Libby has a cornucopia of prestigious award and honorary degrees from various organizations. He has received the highest basic science award from American Heart and Distinguished Scientist Award from American College, gold medal from European Heart, and numerous other prestigious awards from various organizations. He has traveled all over the world for endowed and named and keynote speakership throughout. With that, we welcome Dr. Libby to as the speaker of JCA webinar series under Eugene Braunwald, MD lectureship. Today's panelists are Matthias Nahrendorf, Yibin Wang, Christy Valentine, and Klaus Levy, who will be joining us shortly. Dr. Libby, we are most honored to host you, sir. Thank you, sir. Please.
Well, I'm the one that's honored, and particularly because this lectureship is named after Dr. Braunwald, who was my professional father and has been my mentor and continues to be my mentor today. We were just speaking yesterday about plans for the future of his textbook, Heart Disease. These are my competing interests. This says that I consult widely, but I note that I do not accept any personal payment from any pharmaceutical or device company that markets a product. Well, Valentin Fuster, who's a real hero of our field, invited me a few years ago to write a piece trying to put atherosclerosis in some historical perspective. I'd like to start my talk by rehearsing what we went through in this paper. Let's start hundreds of years ago, when there was a pitched battle between Rudolf Virchow and von Rokitansky about the pathogenesis of atherosclerosis. Virchow, on the basis of careful observation, had deduced that atherosclerosis actually had some aspects of inflammation, and he called it an Entzündungsreaktion, very clearly in the middle of the 19th century. Von Rokitansky took another view, and he thought that the atheroma arose by incorporation of thrombi. So that was the hot debate of the mid-19th century. Well, then the era of chemistry arose, and it was found that cholesterol is a component of the atherosclerotic plaque by Otto Windhaus, and that you could feed animals a diet enriched in cholesterol and saturated fat. This was in rabbits by Anichkov and Cholatov. And so the focus for many decades was on atherosclerosis as a lipid storage disease. Then there was the advent of the era of cell biology. The role of LDL, low density lipoprotein, when the ultracentrifuge was introduced, was very clear. There's observational epidemiology that shows that LDL is permissive and probably causal for human atherosclerosis based on human genetics and interventional studies. But all of those observations don't tell us how LDL is promoting atherosclerosis, and that's where I think we're headed here. When the culture of cells became possible in homogeneous populations, the late Russell Ross was one of the champions, and his view was that atherosclerosis was caused by a bland, smooth muscle proliferation, and he was joined by his mentor, Earl Bendit, in that concept. So this is from the original ideas that Russell Ross and John Glomps had about the pathogenesis of atherosclerosis. There's endothelial denuding injury. There are platelets that will release their granular contents, including platelet-derived growth factor, that will cause the smooth muscle cells to migrate from the media into the intima, as you see here, and to proliferate there. And what's amazing about this construct, which Dr. Ross did eventually revise, was that there's no inflammation whatsoever. So he skated past Virchow's observations from over 100 years before and considered atherosclerosis a bland process. Well, in the 1980s, the tools became available, particularly monoclonal antibodies for immunohistochemical staining, that allowed us to really nail what the cells were that were participating in atherosclerosis. And I'd like to take you on a little walk through memory lane here. Johan Hansen and I, who've been talking about this and working together for many decades, were at a meeting of the Scandinavian Atherosclerosis Society. And we were sitting around the fireplace, perhaps with a glass of wine at the end of a busy and informative day. And someone came by and said, "So, you guys, what's new in this business of immunity and inflammation in atherosclerosis?" Well, that spurred us to write a review that we submitted to the Journal of Clinical Investigation. And as Johan reminded me, we got a letter back from the editor-in-chief without review saying that this looked very nice, but it was not of sufficient interest, general interest, to merit consideration for publication in JCI. So we published it in this pathology journal. And if you look at the table of contents, it's actually strikingly modern, even though it was published more than 3 decades ago. I'll give you a second. I'm not going to read this, but some of the questions that we asked back then have been answered in part, and some of the other questions we're still struggling with. I do want to make a very important point that I already alluded to, and that is that there's not a competition between traditional risk factors such as LDL and inflammatory and immune pathways. The idea is that inflammation provides a series of pathways that can transduce the effects of some of these traditional risk factors and provide us a mechanistic explanation of how they're altering the biology of the artery wall and leading to the disease. So I'm not going to go through this with you. Everyone has cartoons like this in their own collection. When we transition from the normal human arterial wall, welcome these visitors, perhaps unwelcome visitors, to the intima, the leukocytes, about which we'll have something to say in a moment. Then there's the phase of progression of the disease, when indeed we have some of those processes that Dr. Ross advocated so strongly in the 1970s. And there's also cell death that goes on in the advanced atherosclerotic plaque. And then ultimately, disruption of the plaque causes some of the clinical complications that bring the patients to our attention as clinicians most dramatically. In previous lectures, I'm sure many of you have heard me talk about the almost quarter century of work that we did taking apart the cell and molecular mechanisms of plaque rupture, which is a common cause, the most common cause of coronary thrombosis. And the story that we told is that when we have inflammation in the intima, as depicted here by the blue T lymphocytes, if you will, and the yellow macrophage foam cells, that we have a perfect storm for thrombotic complication of atherosclerosis. The T cells secrete gamma interferon that inhibits the production of new collagen required to repair and maintain that all-important fibrous cap extracellular matrix that is all that stands between many of our patients and acute coronary syndromes. We also spend a great deal of time working out the inflammatory conversations, perhaps between T cells and macrophages as shown here, that cause the inflammatory cells to elaborate large quantities of specialized enzymes, members of the matrix metalloproteinase family by and large, that are specialized in cleaving these very stable extracellular macromolecules such as collagen, interstitial collagen. So when we have inflammation, we put the repair of the fibrous cap in jeopardy, and we boost the mechanisms that can dissolve that extracellular matrix and render a plaque friable and susceptible to rupture. Over the last 6 or 8 years, my laboratory has turned to try to understand the mechanisms of superficial erosion, which accounts these days for about a quarter of acute coronary syndromes. And we've worked out some mechanisms, as have others. And we think also that healing of the atherosclerotic plaque, once it's disrupted, is very important in the progression of the disease. We think that there are quite distinct mechanisms for superficial erosion and plaque rupture. I've already shown my hand that we think that the macrophage, the mononuclear phagocyte, is very important in plaque rupture. We have now implicated the polymorphonuclear leukocyte and neutrophil extracellular traps in superficial erosion. So stay tuned. This is a pretty hot topic now in acute coronary syndrome pathophysiology. Well, this is from the late 2000s and shows some of the denizens of the atherosclerotic plaque that we had characterized at that point. And of course, now in the era of single-cell RNA sequencing, I couldn't possibly incorporate on one slide any comprehensible listing of the different classes of leukocytes that there are. But certainly those are what I referred to as the unwelcome visitors to the arterial wall, and that are key in playing out these inflammatory pathways that we think link risk factors to the progression and complication of atherosclerosis. So what contributes to the accumulation of these leukocytes? Is it recruitment from blood? Is it local proliferation within the plaques? Is it extramedullary hematopoiesis or hematopoiesis in the bone marrow? And it turns out it's all of the above. Now, I don't have time to go into all of this, and perhaps you should invite Matthias Narendorf, who's a panelist here, and Philip Swirsky to talk about some of the issues here, which they have really illuminated in recent years. And what is it that triggers the accumulation of leukocytes in the atherosclerotic plaque? Well, there's a whole laundry list of traditional and non-traditional or emerging risk factors. And the answer, of course, is all of these are associated with attracting or causing the accumulation of leukocytes in the atherosclerotic plaque. What are the messengers that cause this recruitment or expansion of the population of the leukocytes? Well, there's cytokines that are protein mediators of inflammation and immunity. There are lipid mediators that are very important and that also may play a role in the resolution of inflammation and healing. And they're reactive oxygen species. Now, I certainly can't go into all of these categories, but I would like to stick to where I think we've had the most translational success, because unfortunately, targeting oxidative metabolism has not yielded clinical results so far. So I'm going to talk about cytokines. Now, back in that paper that we wrote in response to a jocular challenge many years ago, and it was rejected without review by the Journal of Clinical Investigation, we included this table. And if you look at this table, it actually held up pretty well in the ensuing 3 decades. So the conversations about cells of the atherosclerotic plaque, both the intrinsic cells, endothelial cells and smooth muscle cells, and the leukocytes, those of innate immunity and of adaptive immunity, and the players that they secrete and that alter the biology of the atheroma, is actually pretty good. Not complete, but we got some of this right. And that was on the basis of work that we were conducting in our laboratories. back in 1986, quite contrary to immunological dogma, we showed that human vascular cells, endothelial cells are shown here, but also smooth muscle cells could express the messenger RNA that encodes interleukin-1 beta. This caused quite a bit of ruckus, where the immunologists thought that non-properly pedigreed immunologic cells were not capable of producing these wonderful mediators that were called interleukins. Their very name suggests that they're limited to signaling between leukocytes. We also showed, by the way, in this original paper in 1986, the biological activity in the protein synthesis and also IL-1 alpha, as well as IL-1 beta. And you'll note that we had to induce the expression of these inflammatory mediators. In the basal state, they did not express these messenger RNA that encoded IL-1 beta or alpha. And we used what we thought was an artifice in the laboratory, bacterial endotoxin, gram-negative bacterial endotoxin, lipopolysaccharide as a stimulus. Of course, now with our increased appreciation of the microbiome and of impaired epithelial barrier function in certain conditions, LPS seems much more relevant pathophysiologically than we had conceived in the mid-1980s. Well, we very soon after that discovered that IL-1 can induce its own gene expression, and this provided an amplification loop. And we showed, together with Charles DiNarello, who is the godfather of the interleukin biology field, in vivo in rabbits and in human mononuclear phagocytes, that IL-1 can induce its own gene expression. We then went on and showed that IL-1 can induce another pro-inflammatory cytokine, about which I'll have a great deal to say during this talk, interleukin-6. Here is another amplification loop. A little bit of IL-1 alpha will beget huge amounts of interleukin-6. This is from smooth muscle cells. It also works with endothelial cells, but in terms of the bulk of production, we think of the intrinsic vascular cells, smooth muscle cells, will win that contest. Note that this is a log-log scale, so this is a huge induction of IL-6. Now, interleukin-1 beta, like many proteins that are pivotal in biological control, requires a proteolytic cleavage to convert it from an inactive precursor to the active mature cytokine. The enzyme that affects that cleavage is caspase-1, originally known as interleukin-1 beta converting enzyme, or ICE. We localized this enzyme to foam cells in the human lesion in the mid-1990s. We then showed in the biochemical laboratory that a pro-inflammatory mediator that we already knew was active in the atherosclerotic plaque could coax the cleavage of the inactive pro-IL-1 beta to the active form in a way that was dependent on the activity of caspase-1. What we didn't know when we were doing those experiments was at the same time, in his laboratory in Lausanne, the late Jörg Scholl showed that caspase-1 is the business end of a supramolecular structure we now call the inflammasome that senses danger, including some disease-relevant stimuli such as cholesterol crystals or hypoxia, and that will activate the production of the mature forms of IL-1 beta and its cousin IL-18. And then we're off and running because, as I've discussed, we have the first amplification loop, we have the second amplification loop, and this is very formally similar to what we see with the coagulation cascade or the complement cascade, where we have a series of tightly regulated steps that control a biological pathway. Why were we interested in IL-6 in the mid-1980s? Because we already knew that it was the switch that turns on the acute phase response in the liver, telling the hepatocyte to divert some of its substrate and energy from housekeeping proteins like albumin and getting ready for emergencies by increasing the production of acute phase reactants, prominently fibrinogen, the precursor of clots, and the major inhibitor of our endogenous fibrinolytic system, plasminogen activator inhibitor 1. Now, these are bad biomarkers for various reasons, but a great biomarker, although not in the causal pathway for inflammation, is C-reactive protein and another pentraxin, serum amyloid A, which the mouse prefers to CRP for those who are murine experimentalists. So what is it that contributes to the accumulation of the leukocytes in the atherosclerotic plaque? Well, I told you it was all of the above, and I'd like to just focus for a bit on hematopoiesis in the bone marrow, which is something that has interested us. And in particular, I have learned a lot about inflammatory signaling in the bone marrow niche from Matthias and Phil. And in recently published experiments, when they isolated bone marrow endothelial cells from animals with experimental atherosclerosis that consumed an atherogenic diet, Western diet, WD, and did the usual kind of volcano plot, they found that interleukin-6 was prominently overexpressed when we compare the atherosclerotic bone marrow endothelial cells to those derived from control animals. And using the fantastic technology of genetic engineering, which is available today, the virtuosos in the Mass General group, although Phil has now moved to Mount Sinai, engineered animals that have inducible inhibition of the expression of interleukin-6 in endothelial cells. And then we're able to functionally implicate IL-6 in aspects of hematopoiesis as the reduction in these precursor cells, the reduction in the proliferation of the precursor cells, and in peripheral blood, a decrease in the pro-inflammatory subset of mononuclear phagocytes, and even the neutrophils decrease. So it looks like there's a global inhibition, reduction in myelopoiesis when IL-6 is deficient in endothelial cells in the bone marrow. So this is the summative figure in this publication that really implicates IL-6 as a key mediator in the bone marrow niche in hematopoiesis, which can then affect atherosclerosis. Well, another way in which we can have an increase in accumulation of myeloid cells is due to what we call clonal hematopoiesis, which is due to somatic or acquired mutations. And in keeping with this being sponsored by an aging journal, this is a brand new field which links aging, cancer, and cardiovascular disease. If you'd like to learn more about it, I would refer you to this review, although there are probably as many reviews about this topic as there are data papers at the present time. So how does this work? Well, with age, hematopoietic stem cells can acquire somatic mutations in a subset of known leukemia driver genes that favor the expansion of clones of mutant cells, and they can accumulate in peripheral blood. And this is indeed a first step on the road to leukemia. Individuals who have more than 2% mutated cells with a variant allele fraction, VAF of 2, without hematologic malignancy are denoted as having clonal hematopoiesis of indeterminate potential, or CHIP. Why indeterminate? Because the transition to acute leukemia requires the acquisition of up to 3 or 4 of these mutations, somatic mutations in known leukemia driver genes, and it actually happens rather uncommonly, less than 1% per year. So most people who have this condition, which we call CHIP for short, will never have a clinical manifestation, at least in terms of transition to a hematologic malignancy. So why should we care about that as cardiovascular scientists and clinicians? Because although the transition to acute leukemia that requires multiple mutations, just a single mutation in a clone can accelerate atherosclerosis and thrombosis and heart failure. And now it's hard to open a journal without seeing something else that's associated with CHIP. There's some controversy about the magnitude of these effects. We think that a lot of that has to do with the population study and with the stringency of calling the mutations. This is a common risk factor, as I'll show you in a moment, but it also is a very potent risk factor. Fully adjusted for all traditional risk factors, if you have CHIP, there's about an 80% increase in your risk of having cardiovascular disease. And that is on the same scale as hypertension and cholesterol, and is outweighed, of course, by age, which is what we're all interested in this particular webinar, and also diabetes. So this is a very potent risk factor, and it's common. As you see, by the time you're 70, and the world literature really pretty much agrees with this, you have at least a 10% chance of having this condition of clonal hematopoiesis. This is from work of Alex Bick and Pradeep Natarajan, two of my brilliant collaborators. And although there's several dozen known leukemia driver genes, only a handful of these leukemia driver genes cause this condition of clonal hematopoiesis, notably DMT3A and TET2. And in particular, TET2 is the one that we think is the most likely associated with cardiovascular disease. So, of course, there are two possibilities, aren't there? One is that these acquired mutations, which come with exposure to environmental mutagens and aging, are just like counting the rings around the trunk of a tree. And since age is that very strong driver of atherosclerotic risk, it's simply a commensal, it's simply true, and unrelated. And that's why we got together with the discoverers of CHIP, Ben Ebert and Sid Jaiswal, and helped with mouse experiments to test the causality. We created mice that had bone marrow cells that had lack of function of TET2 and did the usual atherosclerosis experiment. And lo and behold, found an acceleration of atherosclerosis. And this was conducted really in parallel with work from Ken Walsh's laboratory that was very similar and came up with the same kind of answer. Now, once you've made these animals, you can make monocyte-derived macrophages and expose them to some of our traditional risk factors, and, for example, LDL cholesterol, and perform RNA sequencing. And when we did that with Sid, we found our frenemies, IL-1 beta, and right downstream of IL-1 beta, as I showed you, IL-6 are overexpressed by these mutant leukocytes. But that's in mice. What about in humans? Well, again, Alex and Pradeep and their team looked in TopMed, the NIH consortium of trials with pooled data, and found that in TET2 clonal hematopoiesis that IL-1 beta and IL-6 were overexpressed. Moreover, it looks like the IL-6 is functional because there's a common variant. 40% of you on this Zoom have a variant which attenuates IL-6 signaling. And if you look in people who do not have CHIP, the cumulative event rate is the same, whether or not you have this impaired function of IL-6 signaling. But if you have CHIP, you see you have a much accentuated risk of atherosclerotic events. And there's very compelling data that we have to consider the idea that atherosclerosis can feed back on the bone marrow and augment the leukophoresis and the expansion of these clones. And that's work of Kamila Naxarova, which Matthias knows very well. And this is another dimension of this link between the bone marrow hematopoiesis and atherosclerosis. So what are the mechanisms? Well, both of these mechanisms are actually operating in the human atherosclerotic milieu, and as well as the mice. But do the experimental results in the mice regarding enhanced hematopoiesis and atherosclerosis actually apply to humans? And for that, we're very fortunate that Professor Fuster was able to undertake a community study. Actually, this is in the employees of the Santander Bank in Madrid. And he has created a cohort known as PESA, the progression of early subclinical atherosclerosis, where he's done pan imaging of these volunteers, mostly men, middle-aged, and without obvious cardiovascular disease. And this is turning out to be a goldmine of information. And in one paper that we were happy to publish in the European Heart Journal, they used the bone marrow uptake of labeled fluorodeoxyglucose to examine metabolic activity. Actually, going back to the work of Warburg in the 1930s about increased glucose uptake as a sign of metabolic activity and glycolytic flux. And looking in the lumbar spine, in the frontal plane, and also in the lateral plane, you see greater uptake of glucose in the participants in PESA that had the constellation of risk factors that we lump in the metabolic syndrome compared to controls. And actually, this technique was something that was worked out in part by Matthias Nerendorf and Zahi Fayyad, which was adopted by the PESA study. Now, we had the opportunity to comment on this very important study with Matthias and Phil and sort of coordinate the mouse work, which had been championed by Phil and Matthias here, the different things that are lifestyle and risk factor associated, and what was found in this analysis of the PESA study. And so we have very good concordance between the experimental group and the human observations of a link between risk factors and hematopoiesis. Well, this is the Bromwald lecture, which is a particular honor for me. And if we go back again 30 years, well, Johan and I were voices crying out in the wilderness about inflammation in atherosclerosis. Professor Bromwald and Victor Zhao got together and put together this cardiovascular continuum. This is the original cardiovascular continuum, black and white, simple slide, showing we start out with risk factors, develop atherosclerosis, develop infarct. We have loss of muscle, we have remodeling, ventricular enlargement, heart failure, and what we now call advanced heart disease. We don't tell our patients they have end stage. Well, with Phil and Matthias, we've been able to flesh out the cardiovascular continuum, bring it into the 21st century. And really, this neural axis is something that's a hot topic now, and that is being expanded, particularly in Phil's lab now. And the idea that there's activation of the bone marrow, that the spleen can be a way station for some of these cells, and extramedullary hematopoiesis can take place there. And we can have a feedback between the consequences of atherosclerosis and more atherosclerosis. And this is a concept which has really caught fire these days. Okay, so this is the pivot point in this talk, because if we go into Google Scholar, it takes us 80 milliseconds to come up with 1.8 million results when we cross inflammation and atherosclerosis. And likewise, immunology and atherosclerosis, 80 milliseconds to come up with over 300,000 hits. So there's a tremendous amount of forests have been sacrificed in the experimental literature and biomarker data in humans. But how do we cross the valley of death from our experimental work and biomarker studies in humans to actual clinical events? So I have, as AJ suggested in the introduction, devoted much of the last 15, perhaps 20 years to trying to move forward from the test tube, from the animal lab into the clinic, and test the hypothesis that targeted anti-inflammatory therapy can improve cardiovascular outcomes in humans. This study was already alluded to. The pivotal study in this field was the canakinumab anti-inflammatory thrombosis outcome study, or CANTOS. Those who wish to delve into the background even deeper than I've already presented, I refer you to this paper. So we designed CANTOS to test the inflammation hypothesis in humans who had survived a myocardial infarction, who were on a full panel of contemporary standard of care secondary prevention, but still had a little bit of inflammation as indicated by an elevation above median for the population in C-reactive protein, that index of inflammation. And we initially were going to have 17,000 patients in the study. Due to business reasons, the sponsor trimmed it to 10,000. You already heard from AJ that the primary endpoint was met. And here in this pre-specified analysis, looking at people who responded best to the intervention, the antibody canakinumab that targets IL-1 beta, by reducing their C-reactive protein below median, you see that there was a greater than 30% decrease in cardiovascular mortality and in all-cause mortality, the holy grail of clinical trials. Will all anti-inflammatory agents improve cardiovascular outcomes? Well, Dr. Ritker, my clinical partner, designed and conceived and got funded the CIRT, Cardiovascular Inflammation Reduction Trial, which used an intervention which has transformed the treatment of rheumatoid arthritis and other inflammatory diseases, weekly low-dose methotrexate. And as you see, the study was a complete null. But if you compare CANTOS to the population that was enrolled in the CIRT trial, the intervention did not alter the biomarkers of inflammation, mediators of inflammation, which were decreased in the CANTOS trial. So in the population studied, methotrexate did not provide benefit and did not actually have an anti-inflammatory effect that we can gauge by biomarkers in that population. Fortunately, we do have a drug that's off the shelf that we can use off-label for cardiovascular endpoints, and that is colchicine, which is an inhibitor of granulocyte microtubular function. And here in the COLCOP trial, led by Jean-Claude Tardif, you see in patients who are soon after myocardial infarction, treatment with colchicine provides a remarkable decrease in recurrent cardiovascular events. And an independent study that looked at patients who were further along in the more chronic phase following myocardial infarction, colchicine in low doses was able to decrease recurrent events. Now, colchicine is associated with a signal for infection, like we saw in the CANTOS trial, and it also is a drug that we wish to avoid in our patients with chronic kidney disease, which really is a large part of the population that we see in secondary prevention these days. So beyond CANTOS, we have colchicine. We don't know really how colchicine works, but it seems to reduce events in certain populations. We have inflammasome inhibitors, and there are at least a dozen programs in pharma and biotech that are targeting the inflammasome upstream of IL-1 beta. And then there's the IL-6 pathway, which I've indicated is causal in aspects of experimental atherosclerosis. Dr. Ritker, more than 20 years ago, showed that your blood level of interleukin-6 correlates with your risk of a first ever cardiovascular event. And there are probably 24 studies that replicate this original study of Dr. Ritker's. We have very strong human genetic evidence that IL-6 is causal. Remember that variant that 40% of you had? If we look in a population of people who have these variants that decrease the function of IL-6 signaling, there is a decrease in the biomarker of inflammation in a gene dosage-dependent fashion, and also a decrease in coronary heart disease. So this is extremely strong human genetic evidence for causality of IL-6 in human atherosclerosis. And indeed, IL-1 beta inhibition may work because it blocks that induction of IL-6 that we demonstrated in my laboratory back in the 1980s. Well, we've mined the CANTOS trial to provide us some stepping stones for the future. And we found that the residual inflammatory risk in CANTOS is associated with IL-6 but reduction, but not with IL-18, which is the cousin of IL-1 beta. And we showed that the outcomes in CANTOS related very well to the efficacy of interleukin-6 reduction. As you see here, those who got below the median IL-6 derived a great deal of benefit in CANTOS compared to those who were treated with canakinumab, but this is in the broken blue line, did not achieve below median IL-6. So where do we go from here? Well, I talked about how colchicine is something we don't necessarily want to use in our very numerous patients with chronic kidney disease. So together with a couple of wonderful nephrologists, we went back into CANTOS and asked, what is it that drives residual risk in our patients with chronic kidney disease? This is very recently published. So the game here was to take patients without chronic kidney disease in CANTOS and those with chronic kidney disease and ask the question, is inflammation or hypercholesterolemia or both that lead to recurrent events. And if we compare the extreme quartiles in CANTOS, those who had the elevation in CRP, the highest levels of CRP, actually had the greatest risk of having recurrent events. But the relationship between LDL and recurrent events in the chronic kidney disease population was null. So it looks like inflammation is really causing much more mischief in the patients with chronic kidney disease than LDL. And that is congruent with what we see from the large-scale clinical trials, that the CKD population or advanced kidney disease population is one of the only places where statins have not proven efficacious in reducing cardiovascular events in the studies like the 4D study and Aurora. So that set the stage for targeting interleukin-6. And we conducted a phase 2 trial, actually during the pandemic, with an antibody that selectively blocks interleukin-6 ligand, ziltivekimab. And we looked at just those patients in whom we don't want to use colchicine, those with chronic kidney disease, stage 3 through 5. This was a dose-ranging phase 2 study, and we found a striking reduction in CRP, which makes sense to you from what I explained about the pathway early in this talk. In CANTOS, we got about a 36% reduction in CRP. We had, with the highest dose of ziltivekimab used in this phase 2 study, an over 90% reduction in CRP. It was sustained. There were concordant changes in a variety of other biomarkers reported in this Lancet study. So that set the stage for another large-scale phase 3 outcome trial targeting the same population. Why? Chronic kidney disease, because it's a large unmet need. It's a very high cardiovascular risk. I'll be on the cardiovascular consult service as the attending physician next week. And I bet you that half the patients I see will have an impaired renal function and will be at elevated cardiovascular risk in association with that disease. And it's a particular biologic state, as we showed. LDL cholesterol seems to be less relevant for outcomes, and inflammation is more relevant. And colchicine, which we can have off the shelf, is relatively contraindicated in that population. So where are we today? In the clinical translation of atherosclerosis, perhaps where Winston Churchill said they were at a turning point in the Second War. Now, this is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. I'd like to thank you so much for the honor of giving this Bromold lecture. And here is a picture of some of the current denizens in our laboratory, finishing a large experiment in times of COVID. Here are some of their names. And again, I want to thank my collaborators and people in my lab for all of the work that made this possible. So thank you very much.
Thank you, Dr. Libby, for this fantastic presentation. I tell you, it must be one of the most gratifying experiences in life to see years of research, basic fundamental research in cell culture, in model organisms, ultimately lead to clinical trial with meaningful clinical impact. Seldom happens in the life of many investigators. And for that, I congratulate you. It's a beautiful journey. Beautiful discovery for years of hard work and fantastic. With that, the panelists, let's start with the panelists. We are open to questions. And if you should be able to unmute yourself, if not, the office will unmute yourself for your questions and discussions and comments that you may have.
I have a question, if I may start. This is Matthias, and feel free to switch on my video also. I somehow can't do that. Peter, that was beautiful. It was a great overview. I really enjoyed it, and I always, still learn something when you give a talk like this. It's amazing. I have a specific question about macrophages in the plaque and also in arteries. You mentioned that now with single cell RNA-seq there is a multitude of subsets and new cell phenotypes to look at. I wonder what your feeling is. What is a reasonable resolution? I'm struggling with this. So, we used to think about macrophages as one cell class. They're not lymphocytes, they're not neutrophils. Then we started to think about recruited versus resident. But now I think, you can play that game until you have, 20 macrophage subsets. And there is probably a reasonable number that's higher than 2. I wonder what you think about this.
So I think with single cell, we're in a very descriptive stage. And I wrote a review with Joran where I put in a paragraph where I said that sort of like Lord Rutherford said, in science, there's physics and all the rest is stamp collecting. I think we're in the stamp collecting phase of single cell. And I evoked the pitched battles among the academics in the Middle Ages, who in the scholasticism school spent years debating how many angels can dance on the head of a pin. And I think that that's where we are. Now, Joran read this draft, and he told me that that was too controversial and flagrant, and that I should remove it, which I did. But then when the paper came back for revision, I put it back in, and he didn't notice. So it's actually in print. So that's what I think. I think we're in a descriptive phase. Everyone is playing with the fun tools. It's the fad these days. It's sort of like if you have a seahorse device, everything is metabolic. And there's a 10x machine, so now everyone can do their beautiful UMAP plots and describe all of the populations that they get. And you're right, it's a game of infinitesimal calculus. The challenge is really to get serious about function and to test in critical and rigorous ways what really makes a difference. So this debate about the angels dancing on the head of the pin is where we are, and that's what your question really throws into stark relief. And so that's a challenge for the field. We need to move past simple categorization and get to function. I think some of the people on this panel actually are working in that direction.
Great, thank you. I think Dr. Klaus, do you have a question, sir, a comment?
Yeah, this is Klaus Ley. Hey, Peter, a great lecture. Can I get you to look into your crystal ball? And we know that colchicine works. We know that canakinumab works. Probably IL-6 antibodies will work. Is there room for combinations? Where do you see this field of anti-inflammatory therapy going?
Well, I think if we're going to combine, we should look at targeting orthogonal pathways, right? So perhaps colchicine is working at the level of the granulocyte, and some of the proteomic data from LODOCO2 suggests that although there's this rumor that colchicine inhibits the inflammasome, the data are really not that rigorous. And the proteomic data from LODOCO2 suggests that it's working on granulocytes. And as I said, in granulocytes, we think that they may be participating in some forms of acute coronary syndrome. So I guess we need, if we're going to combine anti-inflammatory and immune interventions, we should do it orthogonally rather than pile on the same. The idea of targeting IL-6 is that by going one step distal to interleukin-1 beta, we can leave interleukin-1 beta's role in host defenses intact, but attack really what we think is causal in human atherothrombosis. The other way around this is, of course, to target more precisely. And when I give a full lecture about clonal hematopoiesis, I show the landscape of human leukemia and the genetic defects which have been identified through the decades. And I have a big arrow about 22 years ago when imatinib, Gleevec, was approved on the basis of a genetic target. And where are we in the vascular field today with genetically targeted therapies? Zero, okay? Maybe in amyloidosis in cardiovascular medicine, we have some genetically informed therapies. But in the vascular atherosclerosis field, we have nil. So we're more than 2 decades behind oncology. And perhaps things like CHIP will allow us to identify people who will respond to particular interventions. Perhaps in JAK2, V617F, which has a very elevated cardiovascular risk, perhaps we can use a JAK inhibitor. In TET2, we may be able to use an inflammasome IL-1 beta or IL-6 inhibitor, and down the line. So I think that number one, I would use orthogonal pathways if I'm going to combine agents. And number two, I'd like to really stop talking about the buzzword of precision medicine and get there. And certainly many people are working towards that end, including our group.
In that spirit of better targeting and more specific, you didn't mention the adaptive immune response. Do you think the autoimmune response to ApoB and other atherosclerosis antigens plays a role?
Well, you're going to tell us. I mean, your group is very far along, and you know, it's interesting. You started with the tetramers, and now the tetramers are out of fashion. You've superseded that, but your group is probably one of the farthest along in the clinical translation, or we're preparing for the clinical translation of targeting adaptive immunity. And of course, there are other groups as well at UCLA and in Malmö, and so we haven't got the clinical fruit yet, but I certainly do believe in the operation of adaptive immunity. As I said very early on, I think the T cell mediators, and I've written about this, is I think the T cells are the generals and the mononuclear phagocytes are the infantry, that the mononuclear phagocytes are the orchestra members, but that the T cells are the conductors, or the T cell committee get together to orchestrate the innate immune response. So I am definitely a believer in causality of adaptive immunity, and some of the early work that we did in transplantation arteriosclerosis, I think bore that out, where we were in one of the very first experiments with knockouts, we were able to show that gamma interferon is causal in experimental allograft arteriopathy, but not in the mesenchymal, in the parenchymal rejection in allografting. So I definitely have believed for decades in the importance of the adaptive immune response, and wish you well, Godspeed in your effort. Please do it in time for me, all right?
Peter, question about CHIP.
Oh, Chris Valentine, yeah.
So, we tend to lump this together, but as you just mentioned, DMT3A is the most common observed variant in terms of the driver gene, but it really has a very, in fact, the more you look at the data, the weaker it appears the association with events. And so TET2, ASXL1, JAK2. So we're probably going to have to, and then when they looked at the epigenetic changes, they were in the opposite direction. DMT3A was very, very different from TET2 with this. So I guess this is the next step when you talk about precision. We're going to have to look at each of these things, huge data sets, but looking at probably a lot more detail at the specific driver mutations to understand fully what's going on.
Yeah. So what you're saying is if you see one chip, you see one chip. And Christy, we have a paper in press together that is suggesting that there's a particular diathesis towards increased peripheral artery disease in the DNA damage and repair, these such as PPM1D and TP53. And so that's wonderful work that we enlisted you in, and I thought
Derek's paper is very impressive. That it's not uniform, and we'll have to look at these probably a little bit more carefully, don't you think?
Yeah. So that paper is now in press, but it's available in preprint form. Derek Claren led that, and you participated in it, and I think it's very, very informative and very interesting. That there's a predilection towards PAD with the DNA damage and repair mutations. And I agree with you about DMT3A. The AXL1, it turns out that there's a PCR artifact in the genotyping that's been done in UK Biobank. And I think the jury is allowed an AXL1. So there's a very recent paper on the fine points of chip calling by Alex Beck and Pradeep and Sid Jaiswal, that I think some of the differences in the publications about the degree of increase in cardiovascular risk with CHIP mutations can be accounted for because of technical issues and the population.
Right.
Everyone mines UK Biobank, and remember that UK Biobank is predominantly Northern European, almost all Caucasians, and well enough off to volunteer to participate in the building the biobank. And certainly the patients that I see and patients that you see also in your environment don't fit that profile.
Correct.
We see people from more disadvantaged backgrounds and with much greater ethnicity diversity than in UK Biobank. So we all use the UK Biobank, but we should not pray at its altar.
Wonderful, thank you. So we have a couple of questions online. I'll just take one of them. Another distinguished scientist, physician scientist, Dr. Tagmeyer. His question is, fabulous journey. Quick question. What is the effect of diet, intermittent fasting on interleukin-6 and bone marrow transformation?
Yeah, so thank you, Heinrich, who's been a colleague and a wonderful friend for longer than either of us care to admit. And he has the small lab with big ideas and was the last graduate student of Sir Hans Krebs and has wonderful anecdotes about that, which he's written about recently. So thank you, Heinrich. I think that lifestyle is primordial in cardiovascular prevention. It's a battle that we're losing globally. And I think that certainly there's evidence that the Mediterranean diet can lower biomarkers of inflammation. People have not focused specifically on IL-6, but I think that that's a very interesting question. But, I presented a talk at the Italian Society for Cardiology, and the question that came up from the audience was, well, now that we whipped LDL cholesterol, what's causing all of this? And I said, it's the global pandemic of obesity, which is going to be driving cardiovascular risk. And unfortunately, we're sowing the seeds for a whole disaster because of the obesity in childhood with concomitant dysmetabolism. So I'm very concerned, and I think that diet and physical activity are the foundation of cardiovascular prevention, particularly in youth. Although perhaps the application of polygenic risk scores will give us a handle, again, in precision medicine, in who should get more intense intervention in youth. But we're never going to have clinical trials looking at outcomes that in therapies that are allocated on the basis of polygenic risk scores in youth, because no one will fund them and no one will live long enough to see the results. So thank you for that question, Heinrich.
So final question by Dr. Zhilong Jia. Wonderful report. Are there other anti-inflammatory targets for the atherosclerosis except for interleukin-1 and interleukin-6? Thanks.
Yeah. Well, as I said, the inflammasome is being targeted, and there are dozens of cytokines. We did experiments with neutralizing CD40 ligand back in the dark ages. And there are many pro-inflammatory cytokines which can be targeted. The trick, though, is to find those that are causal and those whose neutralization or inhibition will not impair host defenses and will not impair tumor surveillance. When we went to Basel, Dr. Ricker and I, back in 2009, to try to convince the leadership of Novartis, we call them the gnomes of Basel, to undertake this experiment with us. And to their eternal credit, they did. But the questions that came up were, number one, are we going to be reactivating tuberculosis or opportunistic infections? And that turned out not to be the case in CANTOS targeting IL-1 beta. And they were concerned about whether we would be precipitating ventricular rupture or the other mechanical complications of acute coronary syndromes. And in fact, we didn't. And in the era of primary PCI, these complications have become much rarer. And experimental work that we did with Phil and Matthias, neutralizing IL-1 beta, we saw actually improvements in myocardial remodeling, not adverse effects. And so I think that we have to be very careful. You have to get into thousands and thousands of patients studied for many years in order to see these infection signals. And so what we need to do is to really target the causal mediators and be very wary about impairing host defenses. And then tumor surveillance was the other thing that came up in our discussions, or I brought it up in our discussions with Novartis back in February 2009. And of course, as people know, actually we saw a reduction in cancer. And in fact, that is why the study was allowed to continue to completion despite the excess infections, because there was at the highest dose tested a 77% decrease in lung cancer deaths during the course of the trial. So our concerns about impaired tumor surveillance were not germane. And actually, one of the reasons we don't have canakinumab as a drug for cardiovascular medicine is because the sponsor ran off to do a bunch of cancer trials. They were so enamored with the exploratory analysis that we did on cancer. And there's very good biology. I've written about with Sebastian Kobold, who's an oncology colleague from Munich, from LMU. We wrote a piece about the commonality of the tumor microenvironment and the atherosclerotic plaque, and how, although very upstream, we think that cancer is more somatic mutations than atherosclerosis, but we're learning more about the somatic mutations that drive athero. But once you get the disease started, the pathways and the mediators are very similar. So I was not surprised, or I was ready to explain some of the benefits for cancer metastasis on the basis of work that we did on inhibition of metalloproteinase expression by limiting IL-1 beta signaling back in the 1990s. So I think there are many targets, but we have to choose them judiciously.
Thank you very much. With this wonderful webinar, Dr. Eugene Bromwald, MD lectureship comes to its end. I'm grateful to our panelists, Dr. Yibing Wang, Dr. Christy Ballantyne, Dr. Klaus Levy, and Dr. Matthias Narendorf. And Dr. Libby, our sincere gratitude to you, our admiration to you for what you have accomplished in this beautiful journey that you described to do. Thank you all very much. Thank you.
Bye-bye, and thank you.
Thank you very, very much.